Okay, welcome everyone to this new video in cell biology. In this video, I will be summarizing the structure, function, and synthesis of the cell membrane. If you would like more details about these different components, you may refer to the previous videos that I made in this channel. So first of all, the definition of the cell membrane. What is a cell membrane? A cell membrane is a biological membrane which separates the internal environment. So if you look here, it separates the cytoplasm, the internal environment, from the external environment of the cell. In this case, the extracellular matrix. Now, the external environment could be a next cell, for example. So it separates this cell from the next one. It's mainly composed of phospholipid bilayer and proteins, but also has carbs and other types of lipids as well. So if you look here, this portion of the cell membrane is mainly made of lipids, and specifically phospholipid or phospholipids. And it's called phospholipid. That's why it's called phospholipid bilayer. Now, bilayer because we have two layers. We have a layer or inner leaflet that is facing the inside of the cell or facing the cytoplasm, and we have an outer leaflet that is facing the extracellular matrix or the cell membrane of the next cell. Okay, that's why it's a bilayer. Phospholipids or phospholipid, as I said, because most of the lipids here are or belong to the group of phospholipids. There are other types of lipids that I'm going to explain in the next slide as well. There are also proteins, which are major components of the cell membrane, as you can see here. And there are also carbs. So carbs, which are usually attached to the different types of proteins, and specifically to the outer portion of the protein. So it's facing the extracellular matrix or facing the outside of the cell. Now, if you look at the different structures that the cell has or the cell membrane has, you're going to find that there are lipids, as I said, there are proteins, and there are carbs. Now, there are different groups for each of these components. So lipids, we have phospholipids, which are lipids attached to a phosphate group. We have glycolipids, which are lipids attached to carbs, mainly monosaccharides and oligosaccharides. And then we have sterols, which are a subgroup of steroids like testosterone, estrogen, etc. Now, phospholipids make the majority of these lipids. So if you extract all the lipids that make the cell membrane, then probably 50% or even more are phospholipids. That's why it's called phospholipid bilayer. Glycolipids are actually the least common types of lipids in the cell membrane, and sterols are also quite common, probably as common as the phospholipids, because their existence within the cell membrane is really, really important. Now, there are two main groups or subgroups of phospholipids that are known or common within the cell membrane. We have something called glycerophospholipids, and we have something called sphingophospholipids. Now, these are phospholipids that are attached to something called sphingosine, and these are phospholipids that are attached to something called glycerol. Now, glycerophospholipids are the most common phospholipids and the most common lipids overall within the cell membrane. Okay? Now, there are four main types of glycerophospholipids that can be found within the cell membrane. We have phosphatidylcholine, phosphatidylinositol, phosphatidylserine, and phosphatidylethanol-amine. These are the main examples that can be found within the cell membrane, the main glycerophospholipids that can be found. For sphingophospholipids, the main example or the most common example is the sphingomyelin, which is very important within the nervous system because it surrounds some of the neurons that have myelin sheath, hence the name sphingomyelin. Okay, So these phospholipids are the most common types of lipids that can be found within the cell membrane of cells, animal cells specifically. Now, glycolipids, there are two main types. We have glyceroglycolipids and sphingoglycolipids. Now, don't get these confused with the different phospholipids that I just mentioned. So glycerophospholipids are not the same as glyceroglycolipids. Here we have phosphate, here we have a carb, or carbs, glycol. Now, the third main group, or third main type of lipids that can be found within the cell membrane are the sterols. One of the main examples is cholesterol. Okay, cholesterol can be also common to the cell membrane and can be found a lot within the cell membrane. It's probably as much as the different phospholipids because it's important within the cell membrane is really, really significant. Now, these are the different lipids that can be found within the cell membrane. What about proteins? So there are two main classifications of proteins that can be found within the cell membrane. We have something called peripheral membrane proteins, and we have integral membrane proteins. The peripheral membrane proteins are proteins which are not permanently attached to the cell membrane. So they can actually dissociate and leave the cell membrane under certain conditions. The integral membrane proteins are permanently attached to the cell membrane. So they cannot basically detach at any time without disturbing the membrane. So if you want to extract an integral membrane protein, then you have to disturb the cell membrane. You have to basically dissociate the cell membrane. The lipids have to leave, and like there's a mess basically that takes place when you try to extract integral membrane proteins. But in the case of peripheral membrane proteins, you can actually extract them in the lab without the need to disturb the cell membrane. Okay? Now, there are three main classifications of integral membrane proteins. We have something called monotopic integral membrane proteins. These are proteins that penetrate only one leaflet of the cell membrane. So they don't basically penetrate the whole cell membrane. They only penetrate one leaflet, either the outer or the inner leaflet. We have also bitopic integral membrane proteins. These penetrate the whole cell membrane once. So they penetrate both leaflets, but only once. Bitopic integral membrane proteins. Now, polytopic integral membrane proteins, they penetrate the cell membrane more than once. So they could be 3 times, 4 times, 7 times, etc. These two are called transmembrane proteins because, again, they penetrate the entire cell membrane. That's why they're transmembrane. Now, the polytopic, one of the major examples of polytopic membrane proteins or polytopic integral membrane proteins are transport membrane proteins. And these are categorized into two main types. We have channel proteins, which are basically channels. So they're like holes within the cell membrane. And we have carrier proteins, which are carriers. So basically they take whatever substance they're assigned to from one side of the cell membrane and they transfer it to or carry it to the other side of the cell membrane. So from the outside to the inside or vice versa. Now the third type is the carbs. So carbs, it's really hard to classify carbs as an individual structure though, because carbs can be part of proteins or lipids. Okay, but I mentioned it here as an example for the glycocalyx. The glycocalyx is basically all the carbs that surround the cell and the different carbs that have significant functions within the signaling, cell signaling, basically. They have important functions in cell signaling. So these carbs could be part, as I said, of the lipids. So for example, glyceroglycolipids, sphingoglycolipids, or they could be part of the proteins, because most of the proteins that have a region outside the cell, so they have a portion outside the cell facing the extracellular matrix, are actually glycosylated, so they have carbs attached to them. Okay, so this is basically a summary of what I just said. So we have here cholesterol, for example. They exist in both sides, and we have the different types of phospholipids and glycolipids and sterols, like the cholesterol as an example. Okay, so when you look at the cell membrane, it's not only, we're not talking about only phospholipids, but the majority of the lipids that make up the cell membrane are phospholipids. So they're lipids that have a phosphate group. This is also a okay, so here we're looking at the different types of proteins. So we have, as I said, integral proteins, which are mainly these types, and we have peripheral proteins, which are mainly here. So the integral proteins, we said that there is something called monotopic, like in this example, penetrates only one leaflet of the cell, or this one as well. We have bitopic, like this one, that penetrates both leaflets, but only once. And then we have the polytopic that penetrates the entire membrane more than once, like this example here. And then we have the peripheral that are attached, not firmly attached to the cell membrane though. So these can actually leave the cell membrane and go and do some work inside or outside the cell. So the main function of the cell membrane, as I said, the cell membrane separates the internal environment from the external environment. So you don't want everything that's outside to come inside the cell and vice versa. So the main function of the cell membrane, organization of the internal environment. And they do this through different ways or processes. One of the major ones, through organizing transport across the cell membrane. So if we look here, there are different types of transport across the cell membrane. We have something called simple diffusion, which is basically transport through the cell membrane and the lipid bilayer directly, without the need of any help from any proteins. This is called simple diffusion. No energy is needed here. Now, there are certain rules for this simple diffusion through the bilayer. The substance has to be small and polar, okay? Like, for example, water and ethanol. They are small and polar, and they can pass through the cell membrane directly without the need of any proteins, with the exception of water, though, which I'm going to talk about in a few seconds. They have to be small, nonpolar molecules, and these, for example, like oxygen, the different gases, oxygen, carbon dioxide, nitrogen. These are small and they are nonpolar, so they can just go through the cell membrane bilayer without the need of any proteins. Large nonpolar molecules, they can also pass through the lipid bilayer, for example, like steroids, testosterone, estrogen, etc. They can go straight through the lipid bilayer without the need of any help from any protein. But they basically go through different rates. So the small polar molecules, they go by a very slow rate. So they slowly go inside the cell or outside the cell. In the case of small nonpolar molecules like oxygen and carbon dioxide, no, they go really fast. Okay, in a very fast rate. Large nonpolar molecules, they go through very slow rates. So testosterone can be absorbed inside the cell very slowly. But it can go like without the need, as I said, of any proteins. Okay, what about large polar molecules like glucose or some of the amino acids? What about ions like sodium ions, calcium ions, chloride ions, hydrogen ions, potassium ions, etc.? Well, they need proteins or different protein channels or protein carriers, like in this case. So in the case of glucose and amino acids, They go inside the cell or outside the cell, usually using carrier proteins. So the carrier proteins, they take the glucose, for example, from outside the cell and carry it to the inside of the cell. Now, this could happen through either active transport, so by using ATP or using some sort of energy, ATP or otherwise, or by facilitated diffusion without the need of energy. It depends on the type of the channel, basically. Okay? Or, sorry, the type of protein or carrier protein in this case. In the case of ions and the majority of water, they go inside the cell or outside the cell through facilitated diffusion, so they don't need energy, but they need a channel protein. Okay, so they go inside the cell or outside the cell through these channel proteins. Now, as you can see, I said water here and there, because water can go straight through the lipid bilayer without the need of proteins, but it goes through a very slow rate. But the majority of water, it need basically protein channels in order to go inside or outside the cell. And this is the fastest way, basically. And these channels are called aquaporins. Okay? So, the synthesis of the cell membranes proteins. Now, there are two main types of so-called translation, because before translation, there is a process called transcription. Transcription is basically copying the different genes responsible for different proteins. So to sum it up, basically you have genes that are responsible for certain proteins. These genes are copied into structures called messenger RNA or mRNA. These mRNAs are basically the manuscript, or they carry, there are documents that carry the instructions for protein production. The mRNA then travels from the nucleus to the cytoplasm, where it attaches itself to the free ribosomes in the cytoplasm, and the process of translation starts there, which is basically translating these codes or instructions into proteins. Now, there are two different types of translation. Translation, as I said, it starts in the cytoplasm. It can finish completely in the cytoplasm. In this case, it's referred to as cytoplasmic translation. Okay, so it starts here and ends completely in the cytoplasm. Or it can start in the cytoplasm and then pause and continues in the rough endoplasmic reticulum. In this case, it's referred to as co-translational translocation. Co-translational because there are two organelles that are responsible for the translation, ribosomes and endoplasmic reticulum. Translocation because there is a translocation, basically. It moves from the cytoplasm. The process of translation moves from the cytoplasm to the endoplasmic reticulum. Now, this is the most common, basically, type of translation, or the most common type, of translation in eukaryotic cells. There's another type called post-translational translocation, but it's not that common in eukaryotic cells. Now, if you can see here the process, first, the translation process starts in the cytoplasm, where a portion of the protein is produced. After a signal sequence is produced, this signal sequence, basically a series of specific amino acids that send a signal to the cell, so the translation process can stop in the cytoplasm or pause in the cytoplasm and then carry on in the endoplasmic reticulum. So when this signal is produced, a protein that is called signal recognition protocol then attaches itself to the signal and pulls the whole complex, the ribosome, protein, and mRNA complex, to the endoplasmic reticulum. There will be SRP receptor, which is the signal recognition protocol receptor, which will basically adjust the whole complex and attach it firmly to the endoplasmic reticulum membrane. We have a transleucon. Transleucon is basically a hole within the membrane of the endoplasmic reticulum, so the protein can be produced inside the lumen. I previously mentioned an example to make this whole process a little bit simple to understand. So imagine that the proteins are customers. The signal sequence is like the location, and the SRP is like the Uber. Okay, so after the customer sends a location, the Uber comes, attaches or takes the protein, the customer in this case, and reads the location and then takes the customer to the final destination, in this case, the endoplasmic reticulum. Okay, just one last thing to mention. Inside the endoplasmic reticulum is actually outside the cell, or non-cytoplasmic phase. So whatever is produced inside, it will either face the outside of the cell, if the protein is made for the cell membrane. So in this case, this side will face the cytoplasm. If this is made for the cell membrane, this side will face the cytoplasm, and this side will face the outside of the cell. Okay, that's what this means. Now, as I said, when I discussed translation, that translation starts in the cytoplasm using the mRNA, and the ribosome basically is responsible for this process. So to give you another simple example, from a cellular standpoint, ribosomes are like Google Translate or ribosome translate. Now, the mRNA contains the codes or the words that the cell does not understand. Okay, this is the language that the cell does not understand. The proteins are basically the words that the cell understand. So this is the translation or the final product, the proteins that the cell will be using basically at the end. So hopefully this whole thing makes the translation process a little bit easier to understand. Now, the synthesis of the membrane's lipids. So we talked about the structure, the function, how proteins of the cell membrane are synthesized. Now, how are the lipids and the different lipids of the cell membrane are synthesized? So the process starts in the cytoplasm, and this time it involves the smooth endoplasmic reticulum. So rough endoplasmic reticulum, proteins, smooth endoplasmic reticulum, lipids. So the first step is 2 fatty acids, in this case, are bound to something called glycerol phosphate, okay, making something called diacylglycerol phosphate. Okay, this diacylglycerol is called also DAG, which is some sort of important part of the cell membrane, lipids. Okay, so the diacylglycerol phosphate is produced after the reaction takes place between these two. Now, this will basically bind itself to the membrane of the smooth ER. Now, depending on what the cell wants, so depending on the final product, the next step takes place. So if the cell wants phosphatidylcholine, then this phosphate head of diacylglycerol phosphate is replaced with phosphate and choline. This is how you get phosphatidylcholine. If the cell needs phosphatidyl inositol, then this phosphate head is replaced with a phosphate and inositol group. If it needs phosphatidylserine, the same, serine and phosphate will replace the phosphate head of the diacylglycerol phosphate. That's the second step. Now, also in the case of sphingolipids, this glycerol is replaced with sphingosine. Okay, so the steps are usually the same, but the components that are used are different, dependent on the lipid that the cell wants. Now, the third step is the flippase step. So this flips the lipid produced to the, or assign the lipid produced to the appropriate leaflet. So basically, if this lipid is produced for the cell membrane, then if this lipid means the phosphatidylcholine, if this is produced for the cell membrane, then it will be flipped to the inner leaflet, because this leaflet will face the outside of the cell, as I said before. Inside the ER, outside of the cell, or non-cytoplasmic phase. So this basically flipase enzyme will flip whatever product and assign it to the appropriate leaflet, because each type of lipid or each type of phospholipids has a specific leaflet that it works on. So phosphatidylcholine usually found on the outer leaflet of the cell membrane. Phosphatidylinositol and phosphatidylserine usually found in the inner leaflet. So this basically will flip this product and assign it to the appropriate leaflet. Now the fourth and the final step is basically transport of this lipid. So it could be transported through vesicles, for example to the cell membrane, or through a protein called phospholipids exchange protein. So this protein will pick this lipid individually and will take it to the membrane that needs this lipid, for example, the membrane of the mitochondria. So this usually works within the cell where this is, or in this case, it's usually the cell membrane or outside the cell somewhere. So this concludes our video for today. If you like the video and the content of this video and this channel, then please like, share, and subscribe. You may also visit our website for further updates in the future. Thank you very much, and see you in future videos.